It's a comparison of MammaPrint and Oncotype 21 gene assay in performance on 437 samples of early-stage breast cancer at our institution. What we did in this work is compared all of them. We saw which patients, say, were various risk categories with a 21 gene assay, be it low, intermediate, or high. We looked at whether individuals' cancers were either high risk or low risk by the 70 gene MammaPrint assay. And then finally, using the 80 gene Blueprint assay, we determined whether they were intrinsically, the intrinsic subtype was either luminal A, luminal B, HER2-like, or basal. And put all that data together and just compared one thing to the next in this particular trial. And so what we're trying to do, at least initially, with this data is not only compare the 21 gene assay to the 70 gene assay, but actually get a sense of how well the intrinsic subtype actually matched what was predicted based on the histopathology. And these are all very, very important because what we want to do at the end of the day is treat people right. We want to decide what kind of therapy to give them, whether they need chemotherapy and hormonal therapy and trastuzumab, or standard hormonal therapy alone. And I think these are the sort of decisions that oncologists make every day. And these tests theoretically help us figure that out. But we really started this analysis trying to figure out which test did a better job of predicting who was going to relapse and who wasn't. And we'll talk about relapses in a minute. The other thing in this particular study was trying to compare how accurately both tests categorized patients either as low risk, high risk, intermediate risk, and whether they really overlapped or not. The third issue, I think, and I think fairly important issue, was how often did the intrinsic subtype of the patient really match the prediction of the intrinsic subtype by histopathology and by classical pathology. And those were three very important things that we did because I think that there are a number of patients who, by standard estrogen receptor and progesterone receptor staining and Ki67 staining and HER2 staining, you predict that they're one intrinsic subtype when in fact, looking at the downstream expression of the genes, through Blueprint, you're finding a completely different intrinsic subtype. And the real question is, for a lot of people, it's not really answered by this, but should you be treating the patient by the histopathological intrinsic subtype or by the intrinsic subtype predicted by the 80 gene Blueprint assay? I think it's a really, really important and fascinating question.